And hello, microbiology fans. So, we had a little Zoom gathering. I may post it. I'm not sure if it's really all that worthwhile. I also probably will just summarize a bit of what was going on, what we talked about. This weekend, I'll put together a review sheet of sorts, sort of what I think we covered, and some of the lists that you need to begin developing for yourself in terms of, having sort of an organized way of putting all this stuff together in your head. And the exam, as you're saying, will be on Wednesday. It's going to be an hour exam, starting on Wednesday and open till Friday, I think is how we're going to do it. And open notes, open book, open computer, whatever you want to do. Just the point being that if you got to, you're going to have an hour, and so you need to have some familiarity with the information. And so next week is actually going to be a pretty cool discussion set on metabolism and the ideas about how microorganisms extract energy from their environment. And what's really interesting, I think, is to compare the way microorganisms are extracting energy from their environment and the way we are extracting energy from our environment. And one of the things that makes it a little bit interesting topic is that bacteria have mechanisms that are so fundamentally different than what we do that it's really a very interesting area of study, in that we get our energy and the components we need from eating. So just to refresh this as a point, because I don't want to kind of jump into the next chapter and confuzzle you about what's going to be going on in this exam, but next week we're going to get into metabolism. But let's just think about what it means to actually consume, and why do you need to eat? Well, you need to have a carbon source in order to build these macromolecules. I mean, carbohydrates, proteins, fatty acids, nucleic acids, they all contain carbon, so you got to consume the carbon and other what we refer to as macronutrients. And these are the different kinds of elements that you need to ingest in order to build all these macromolecules. So obviously carbon, and generally you're going to be consuming hydrogen along with it. Carbon, hydrogen, and oxygen. I mean, these are things that are going to be coming in your carbohydrates. Nitrogen, just as a little check, what biopolymers use nitrogen? And the answer, of course, is amino acids, proteins do, and nucleic acids. So you got to have nitrogen as well. Then we've got a range of other molecules that we use in lower concentrations. Sulfur is a component of the amino acids cysteine and methionine. Phosphorus, of course, is part of adenosine triphosphate, all the nucleotide triphosphates, and we're going to be using phosphate because of its negative charge to drive chemical reactions. Additionally, we've got a variety of other molecules that are elements that are also important for not necessarily building these different biopolymers, but in terms of regulation. Iron, potassium, magnesium, calcium. I would think that chloride also should be in here. These are elements that are being used as part of the salt concentration. Iron is actually a cofactor in a variety of molecules. That is, it fits into a protein or other kinds of molecular structures and serves as sort of a scaffolding thing. And same with a lot of other different elements. So we then have a class of nutrients that are referred to as micronutrients. And these are things that you need in really, really small amounts, trace amounts. And for us, if you're eating food, heck, if you're probably drinking tap water, you're getting a fair amount of these things. Weird stuff like cobalt, copper, manganese, molybdenum, nickel, zinc. These are going to be involved as well in various kinds of cofactors and chemical reactions that you have to have. So you don't need much of them, and it's pretty easy to maintain them, but you do have to have them. And particularly when we start thinking about bacteria growing in really weird environments, they may not have necessarily all these different nutrients and things. There are a variety of other organic chemicals that you have to have for growth, and a lot of pathogens require these. So these are some molecules that we'll be getting into. You don't need to really memorize this. NAD, vitamin K, things that you can't biosynthesize. Sometimes there are organisms that can't biosynthesize all of their amino acids, so they have to take in amino acids. You often have to get these from the environment or maybe from a host. And one of the things is that some esoteric, weird-ass bacteria, they can really be hard to culture in the lab because you don't necessarily know what their requirements are, and they may need some sort of weird molecule that isn't standard in the lab shelf or the lab media. So these are some of the different things we talk about. Now, in the lab, we have a whole range of culture medias, and we can kind of break them down into two different types. One is called a rich medium or a complex medium. And this is going to be a medium that is complete. It has all the necessary requirements, and it is a full meal. And so one of the things that we do for this is rather than adding piecemeal all the little bits and pieces, you typically take some kind of dead living organism like yeast and digest it a bit, crack it open. In fact, a standard thing in the beer brewing industry, there is what's known as brewer's yeast, or you can find it at the health food store as nutritional yeast. And this is dead yeast. Eating live yeast would give you a bit of a bellyache. But dead yeast comes from the results of doing beer brewing, and there's a lot of that. And so you take the yeast at the bottom of the fermentation tank, of the beer tank, and you essentially cook it, blow it apart so it's not in its cell walls. And because it's a living organism, it's going to have everything. It's going to have all the different kinds of macro and micronutrients, or it was a living organism. It's just a really useful food, both for you and I and for the bacteria. We often use things like milk protein. You can break it down. Also, even using different kinds of tissues like there's a component in the lab that's called bactotryptone, which is basically a very dense dehydrated beef broth. So anytime you take material from living creatures, that's probably going to be a really complete and rich media. One thing is, it is a bit of a black box because they can tell you sort of the nutritional breakdown, but you don't know everything that's in it, and it generally doesn't make any difference. Sometimes we use what is known as an enriched media. This would include certain weird things like sometimes there's blood as a requirement for certain microorganisms to grow on. Other times you can add extra sugars just to give the bacteria a good bump and make them grow faster and more robustly. Another media is known as defined media, and this is when you actually take every single amino acid and every single kind of component required for living systems, a nitrogen source, different carbon sources, and you physically weigh them out and add it all in, rather than using a sort of unknown complex media. And so this is often done for very specific reasons of culturing bacteria under different nutrient conditions and things. Other media we have is things that are called selective media that will allow one strain of bacteria to grow and inhibit another strain of bacteria. And
what's that?
I have somebody in the lab holds it. It'll give me about 15 minutes. So, selective media. And one standard selection is increasing the salt concentration can inhibit certain bacteria and support the growth of other bacteria. Different sugars, some bacteria can't use those sugars. A variety of different kinds of things like pH. You can actually select for different bacteria. Other kind of media is one called differential media, and I don't expect you to memorize this list. This is just a way of thinking about things. Differential media may give you a color change reaction or a halo around different kinds of bacteria and allows you to identify bacteria this way. So that's some of the nutrition stuff. The last bit, just to mention here, is that we were talking about, and let me see, we'll kill this and more light. When we were thinking about the nutrient requirements and the way we can categorize the bacteria in terms of the nutrition options, and so this is back up in the PowerPoint presentation, we've got a set of things you need in order to carry out life. And we'll be getting into this in metabolism next week. You and I get these things just from eating, and you do it every day, and so you don't really think about it. But plants are not eating. So plants get their source of energy from sunlight. Now, that's a standard kind of alternative energy source, is being a phototroph, getting energy from sunlight. Chemotrophs are getting their energy from breaking apart chemical bonds, kind of like setting fire to something. Another part of the nutrition aspect is we have to have a source of electrons. And this is not necessarily obvious as to why you need electrons, but we'll be getting into this next week as well. You may or may not be familiar with the notion of an oxidation-reduction reaction. So when we're breaking stuff down, we're oxidizing things. When we're building stuff up, we're carrying out reduction reactions. Oxidation is a loss of electrons. Reduction is a gain of electrons. If you're going to be building new macromolecules, you need to actually have a source of electrons to build those bonds. Now, again, we get that from the food we eat. But if you're a microorganism doing some strange sort of nutrition things, you may get that electron source actually from inorganic chemicals, chemicals that don't contain carbon. Things like iron, hydrogen, or hydrogen sulfide, H2S, ammonia. These are things that microorganisms can extract electrons from. You and I can't. We get our electrons from the same place that we're getting our source of energy, and that is from organic materials. Now, the third thing you need to have is a carbon source. I mean, this is, again, hopefully rather obvious, as we're carbon-based life, and so to build molecules, you got to have carbon coming into the system. We, again, get that carbon coming into the system by eating carbon-based food, which is pretty much everything you're going to eat. Some organisms, though, are able to extract carbon from the atmosphere. That is, they can take carbon dioxide or inorganic carbon. These are autotrophs, and they're able to actually pull carbon in from the atmosphere. They have to have these other micronutrients and things like nitrogen and all these other salts, but they can build organic molecules just from CO2. So again, we're heterotrophs. We are consuming large macromolecules. You can begin to start playing around with a weird diet where you ate nothing but glucose, pixie sticks, and that would do you for a while. You are going to need a source of nitrogen, and because we can't biosynthesize all our amino acids, you're going to need a few select amino acids that we can't biosynthesize. So you could go on my famous pixie stick diet, where you really are just getting all your calories from pixie sticks, and every third day you have a piece of chicken. That'll do you just fine. Hey, that's all I know. I'll be putting some stuff together and posting it, and we'll be talking soon. We will start on metabolism on Monday, but the exam will be set up for Wednesday. That's all. I'm out of here. Peace.